In this video, we'll walk through the Mouse Genome Informatics CRE portal, found online at www.informatics.jax.org/recombinase. We'll begin with a short introduction to CRElox technology, move into the database and use MGI to search for a CRE recombinase expressing mouse using anatomical structure or driver, and then search for a specific CRE recombinase expressing mouse beginning from a publication. In a traditional germline knockout, you eliminate expression or function of a gene across all tissues, organs, and cells. The CRElox genome engineering technology allows researchers to remodel the genome in a tissue-specific manner. And in order to do this, we'll actually take a step back into the basics of biology and remind you that tissues and organ development and maintenance relies on control of gene expression, requiring tissue-specific promoters and tissue-specific gene expression signatures. In order to harness this for remodeling, you actually require two different strains of mouse. One, which is your target gene, which you're hoping to remodel, and the other, which is the CRE recombinase expressor, and that's the enzyme that's actually capable of doing the remodeling. On the left, you see a diagram depicting the mouse carrying a floxed allele. Floxed is flanked by LOXP sites. These are the recombinase recognition sites for the CRE recombinase enzyme. And in MGI, these will be tagged with the allele attribute of conditional ready. On the right, you'll see a diagram describing the CRE expressing mouse, and the CRE recombinase is placed under control of a specific promoter, here indicated with the heart. The allele attribute for these is recombinase in MGI. You take these two strains, cross them together in order to get both alleles within the same mouse, and what you'll see is that in tissues where the CRE recombinase is not expressed, the flox gene isn't touched, it isn't remodeled in any way. However, in tissues where the CRE recombinase is expressed, in this case diagrammed as the heart, it's able to recognize its LOXP recognition sites, remodel the DNA, and in this case excise the intervening sequence, so you have a tissue-specific knockout. We're going to be focusing on the CRE recombinase and promoter activity side of things. To do that, we'll use MGI's CRE portal. So if you start from the home page, you'll see the recombinase button right there. Click that, and you've gone to the CRE portal, so the recombinase or CRE activity mini homepage. On this page, you'll see sections to access data with a nice forms right there on the some frequently asked questions, some statistics, and we'll zoom in just here into the access data. To find recombinase carrying alleles, this allows you to search for tissue recombinase activity assayed in a particular tissue or organ system, or driven by a particular known promoter, or to combine the two. And we'll just click on that and go have a look at how it seems. So, very straightforward here. What you do is you just begin typing. I'll start typing ACTA1. You'll see a drop-down list appears that gives you all of the potential options. I'm actually not going to select one here though. I'm going to use this instead and start typing heart. Again, you see each of the anatomical structures where data annotation to some allele or another occurs. Select heart, and you get the results table. It's a fairly large table. You'll see the allele symbol is here in the second column, with the driver indicated in the first. This is the meat of the column. You've got the recombinase activities detected in these systems, is not detected in those. If this is an inducible allele, it's induced by tamoxifen, doxycycline, or similar. If you're looking to obtain these mice from a public or commercial repository, if that repository is indexed by the International Mouse Strain Resource, or the IMSR, you can get direct links from this column. If this mouse has been used in a publication that we have indexed within MGI, you can see the references associated with it here. So the filters, if we want to ensure that this is not only been assayed but detected in the cardiovascular system, place a check mark in the cardiovascular system, apply that filter, and you'll notice that now cardiovascular system only appears in the recombinase activity detected side of the column. If, for example, we also wanted to ensure that it was not detected in the liver and biliary system, we can place a check mark here using the not detected in system filter and filter that. And now we know that all of the remaining alleles have Cre recombinase activity within the cardiovascular system, not in the liver and biliary system. And we can explore this a lot further by simply taking an example, clicking on it to go to the transgene allele detail page. Here I'll draw your attention to two places: transgene description, which is right here. This includes all the details of the allele's construction and generation along with the original reference. And then the recombinase activity table, which is here. You'll see multiple developmental stages are indicated, which may or may not have data. In this case, we only have data corresponding to the adult. And when the recombinase activity was assayed and detected, a check mark appears. When the recombinase activity was assayed and not detected, a minus or dash appears. You can see that right here. Each of these can be expanded if you click on the small triangles beside to see exactly which tissues have been examined. And these again can be clicked on to go to an even more detailed page that describes the specifics of the assay, describes specifics of the specimen, in this case postnatal week 12 to 14, level is strong, pattern is in this case not specified. For many alleles, not for this particular example, you'll also have images accompanying these annotations. So that's how to use this the Cre portal to find a recombinase by some data type, whether again as the promoter or the tissue assayed in. I'll take a brief aside to say you can go by publication as well. In that case, you'll actually use a different entry form. Go to the search drop-down menu, select the reference query right here, and you can use as much or as little data to enter into this form. Here we'll use the example of the author, Junichi Miyazaki. We'll make sure it's talking about Cre, so we'll use keyword Cre, run the search. You can see we have a number of options. If this is the paper that we were particularly interested in, a transgenic mouse line that expresses Cre recombinase activity in mature oocytes, we see right here phenotypic alleles. Click on that. The one with the recombinase attribute is this one right here, transgene CAG Cre 13 MIA. And once again, we can click on that, retrieve all of the similar information as before. So I hope that answers some of your questions. If you do have any more questions, please do feel free to contact us using the button right here, and we're happy